So, good welcome to the second day of the School of Soil, Biodiversity and Bioindication of the Italian Society of Soil Science. It is an honor for me to start this second full day with a keynote lecture given by Dr. Stefano Mocali, entitled Exploiting the Native Soil Biodiversity to Promote Crop Productivity and Sustainability. Dr. Stefano Mocali is a biologist with a PhD in soil science and climatology, obtained at the University of Florence. Actually, is employed as a researcher at the CREA Agriculture and Environment Research Center in Florence. I can say that Stefano is a brilliant microbiologist with a broad experience in soil microbial ecology and biogeochemistry. Some international fellowships allowed him to improve his knowledge in molecular methods. He was at the Julius Kuhn Institute of the Federal Research Center for Cultivated Plants in Braunschweig, Germany, and at the Netherlands Institute of Ecology in Wageningen. Stefano was chair of the Soil Biology, Microbiology and Biodiversity Committee of the Soil Science System within the European Geoscience Union, and he was also chair of the first division, Soil in Space and Time, of the Italian Society of Soil Science. Actually, he's the Italian Young Ambassador of the International Society of Macrobial Ecology and member of the Earth Macrobiome Project Consortium. And actually, Stefano is coordinating an H2020 project, Excalibur, and I'm sure that this morning he will present this project to us. Please, Stefano, it's your time.
Thank you, Paula. Thank you, Eduardo. I'd like to thank all the organizers for inviting me and giving me the opportunity to have this talk this morning. I'm really glad to be here in the Soil Biodiversity and Bioindication School of the Sister Society. And thanks, of course, for your nice presentation. So, my presentation, I've organized the presentation in this way. First of all, I will briefly introduce some basic concepts related to soil biodiversity and sustainable agricultural management. Then I will present a couple of case studies, and which will be useful to introduce a brief description of the Excalibur project that Paula just mentioned. So I will conclude and I try to give some recommendations. I'm pretty sure that most of you are quite familiar with the soil biodiversity concept, but maybe some of you still is not able to completely realize the importance of the huge amount of soil biota and biodiversity which we have under our feet and is present below ground in both forest and agricultural soils. This huge amount of invisible biodiversity is really important because it's the main actor in the driving force of most of the soil processes and functions, and with a number of key impacts on our lives and on the planet health, of course. And maybe it's not share the information that soils contain about a quarter, more or less, of the all living species of the planet. But we know just about 1% of these organisms. So we essentially we don't know who's there and who's doing what. However, the soil biodiversity is really important issue, and to understand why it's such, it has such importance also at not only in scientific sense, but also for our daily lives. We should keep in mind that soil has a very fundamental role in our lives. Just to remind you that, for example, recently the United Nations just published and released the list of the Sustainable Development Goals in the Agenda 2030, and how you can see, highlighted by the green symbols, about half of such goals depend directly on soil. For example, zero hunger, good health and well-being, affordable clean energy, responsible consumption and production, climate action, of course, life below water, and life on land. So, keeping in mind the centrality of soil, which has to be considered not just an inert matrix, but a real living being, in order to understand its importance, it's really basic, in my opinion, to have clear all the potential ecosystem services that are provided by soil. Of course, soils are really important for the provision of food. It's the main function, maybe, at least for normal citizens. But we have to also consider the important role of soils for carbon sequestration, for water purification and soil contaminant reduction. Of course, the soils are the main actor in regulating the greenhouse gases emissions among soil and atmosphere, regulating the climate changes, and also regulated the soil nutrient cycling. Is, as I mentioned before, is the habitat of for a number of different organisms. It also regulates the water flood regulation below ground. Another important function is represented by its role as a really amazing genetic resource sink. It hosts a number of billions of potential useful molecules for drug development and pharmaceuticals. And of course, soils are also important for human activities like infrastructures. They provide construction materials and also are important for cultural heritage. So I invite you to give a look to these different ecosystem services and to realize that most of these services are driven directly by soil microbial communities. That's amazing. The first time I realized it, I thought, what incredible role plays such small and visible organisms within soil matrix. So now you can have an idea of why soil biota, soil biodiversity is so important for our lives, for our health, and for agriculture, and why it's so important to preserve such biodiversity in terms of genetic biodiversity, but also in functional terms. And yeah, agriculture plays a really important role in shaping the overall soil biodiversity. The question is, and the challenge of the next future is to understand which are the potential impacts of agriculture on soil biodiversity, how it can manage, which is the best way to manage agriculture in a sustainable way in order to combine the crop production for human necessities and the preservation of soil microbial diversity. So it is well established, and maybe you are already aware of the impact of the intensification of agriculture on the soil biodiversity. As this demonstrated several years ago, so it's well known, but a nice publication demonstrated that an intensification of the land use across Europe, and this study was conducted, for example, in Sweden, United Kingdom, Czech Republic, and Greece, they found out that the identification of the land use from grassland throughout extensive and intensive agriculture significantly reduce the soil biodiversity in terms of a number of functional groups, as you can see here, in terms of overall Shannon index, and also average trophic level. So definitely, we know now that intensification of agriculture leads to a loss of biodiversity. So the question is, can we restore, can this biodiversity loss can be restored? The answer is yes. So I would like to show you an example, an experiment that was conducted in this nice experimental site in the Bellewaerde region in the Netherlands, where ex-agricultural fields are available, and these fields are special because they were abandoned. They were abandoned several decades ago over time. So we have a sort of chrono sequence available for assessing the soil biodiversity dynamics over time. And a researcher from a group from Wageningen demonstrated that, for example, when they assess the overall biodiversity in the soils recently abandoned. Recently, it means since at least 5 years. The overall number of species, which is represented by circles, and they accounted for bacteria in red, also fungi, plants, plant feeders, and so on. So well represented here, but the nice result they showed that focused on the network interactions occurring among biotic species. And as you can see, in recent abandoned land, the biotic interaction, the network strength is really open, is poor. The microbial communities and the biotic species are poorly correlated each other, whereas over time you can see that the density of the networks increased during the natural restoration of soils. And the remarkable result, in my opinion, is that the increasing network density corresponds also to with an enhanced efficiency in terms of carbon and nitrogen uptake. And soils with high density networks also provided a fewer number of soil-borne pathogens. So when we address some study for assessing soil biodiversity, it's important, of course, to assess the number of species present in such specific soil, but it's also important to know how they interact with each other. And for example, in nature, what happens in natural soils, for example, during natural plant secondary succession? We can see that, for example, soils in mid-late succession phase are more efficient compared to the soils in the early succession phase. And the question is, so if this happens in nature, how can we get such type of results in agriculture? Well, there are several opportunities and methods available. Of course, we can reduce the tillage of soils. We can also increase the amendment of organic matter of soils. We can use cover crops, reduce the use of pesticides, etc. So these are commonly adopted approaches that we can find in literature and all around. But if we consider that natural soils in the early successions host fast-grow plant species, which can guarantee a high productivity, when we get more efficient soils over time, we observe that soil microbial and biodiversity in general can change, of course, and but increasing the efficiency of soils, but also selecting plants, slow-growing plants, which are more efficient in getting the nutrients out from such soils. So if we aim to move agriculture forward, more sustainable soils, we should also consider this aspect. And the challenge is represented by trying to obtain, to get a similar crop yield from the agricultural soils, but maintaining an overall high biodiversity and functional biodiversity, and also high network interaction strength. So just to summarize this concept, which is very, very basic. So we can say that, summarize that in extensive systems, we have a low crop production, but an high soil biodiversity and network interactions, which leads to higher soil processes, higher soil efficiency, and lower resource inputs and outputs from the system. On the other hand, intensive systems, which are typical of many actual agricultural systems, provide a high productivity in terms of crop productivity, but show a low soil biodiversity, a low internal regulatory processes, and network interactions, thus providing the need of higher resource inputs and also losses. So, aiming to obtain, to reach somehow a sustainable system for our agriculture, we should try to combine somehow the two systems and frameworks, trying to get ideally the higher productivity in terms of crop production, but also maintaining a high soil biodiversity and function. So this is the goal, a challenge, a really, really tough challenge. And the question is, how can we get such results? Are there any approaches available or not? The answer is yes. Actually, a number of different approaches have been proposed. A number of different approaches are currently used, and somehow they are really efficient, sometimes not so efficient. So, but I'm not going to describe all the available approaches, of course, but it's important. I'd like to give you an idea of what can we do to go forward a more sustainable agriculture management. So first of all, we can adopt some methods which can say play as an entry point at two levels. The first level is at plant level. So we can choose and select plants more adapted to a specific soil condition. We can enhance crop diversity in order to, how I mentioned before, to optimize the relationship between such specific crop and that specific soil. But of course, as you can imagine, we have a huge amount of different plant varieties available and a huge number of different soils. So to find out the right combinations, the best combination of plant soil to optimize plant soil interactions is not so easy. Another approach is based on soil management, trying to improve soil management, reducing the tillage, soil tillage, for example, or no tillage at all, like for set-aside farming, but also amending organic matter to soil by composting, mulching, etc. And these methods are commonly used and give interesting results. So first of all, I'd like to show you an example where this kind of approach has been used. We had this experience some years ago in a project where which aim was to recover the soil fertility of degraded soil of a number of vineyards across Europe. And the problem with these vineyards soils were main problem was related to an inadequate soil management practices, and which led the soils to forward a degraded state, and an high mortality of plants, reduced yield, and due, of course, to the abiotic stress that occurred in such fields, such providing a number of critical problems for the farmers. So we consider a number, as I said, of different experimental sites. We asked to the owners of the vineyards to select within the same vineyard a degraded area and a not degraded area. And the degradation definition was, let's say, defined based on the symptoms of soil malfunctioning, or lower grape yield, or higher frequency of grapevine mortality. So we decided to ask the farmers to select this area, because they live within their vineyards. They know very well the potential of their vineyards and of their soils. So the first step was to involve the farmers to select these areas within their vineyards. And it was nice to observe, like, for example, in the farms in the Italian experimental sites, Fontone San Disdagio, it was nice to observe, like, degraded soils indicated with D letter provided and showed a significant lower yield in terms of grape yield for vine plants, compared to not degraded soils, which show about the double value of yields. And also the qualitative results expressed, like in this case, sugar content of the grapes was significantly different. As you can see, the degraded soils show significant higher values. That means that we can have problems with wine production, with the alcoholic content of the wine. So the areas selected within the vineyards provided significant differences in terms of plant and grape productivity, but also in terms of soil nutrient availability. Just report the overall organic carbon and total nitrogen content of the degraded and not degraded soils. And as you can see, there are also differences in these terms. So what we've done was to try to adopt 3 different organic management to recover, to restore such degraded soils. And we try to apply compost and green manure and dry mulching to all the experimental sites I just mentioned. Of course, I just show you some results and not all the results, otherwise I would not have time for it. And as you can see, for example, in the Italian experimental sites, where the 3 treatments, degraded soil with compost, with dry mulching, and with green manure have been adopted, we also consider not degraded soil as control for with a good production, and a degraded soil where the production was the lowest of the vineyard. And the next results were obtained in terms of bacterial diversity, soil diversity, where, as you can see, the green manure treatment shaped the soil, the overall bacterial community, in a similar way to the controlled soils, not degraded, so providing the best production. And the similar result was obtained in the other farm and in the other experimental sites of the project. So green manure treatment was really efficient in recovering the native biodiversity of the soils, and making the soil biodiversity similar to the soils which display the best production, and maybe it may be due to the amount of organic matter provided to soils. But as you can see here, different treatments provided a different amount of organic matter, and the green manure is not the treatment with the highest organic matter amendment, because the compost management provided a higher amount of organic matter. Despite this difference, the best results were obtained by the green manure treatment. So the take-home messages of this first case study is that, yeah, soil bacterial biodiversity of degraded soils may be restored with this approach. The restoration soil bacterial diversity is affected by soil organic matter in terms of quality rather than maybe its total amount. So this is to be considered in general. So another point to take present is that the green manure, is that the best solution in general? So in this case, yes, but we have to consider that green manure, but also other amendment practices and approaches are really complex and variable. So sometimes they work, and sometimes are less efficient, and this is because they are really dependent on the interactions with soil, with the native soil biodiversity, with the environment, with the practices, and so on. And another message I'd like to give you is that when you assess soil biodiversity, it's really important to consider also the functional diversity. So it's important to know, to assess who's there, but also who's doing what happened. This is why I'd like to show you, for example, another case study conducted in another vineyard in Tuscany, where the Broglio Castle, where the Chianti wine was invented, okay, in the heart of Chianti area. And this farm was characterized by a pedological survey in order to divide the entire area in among different soil types. So we find out that the overall area was made of different types of soils, which differed in terms of chemical composition, sometimes in physical and lithological properties, and so on. So we define homogeneous areas characterized by similar soil properties in the area. And this is useful information for the farmer, because they could improve their management practices, focusing specific management strategies for specific areas, a sort of precision agriculture, okay? But when we assess the quality of the wine provided by the vineyards or such areas by using this index, a wine performance of Sangiovese. Sangiovese is the cultivar of vines using these vineyards. We, as expected, we find out, we saw that different soils provided different wine quality. And this is not surprising, of course, because we know that soil terroir is really impacting in the soil, in defining soil, wine aroma and traits. But the interesting results regarded this soil, Lecce 2 area, which was supposed to be homogeneous, provided two different kind of grapes and wine within the same area, within the same vineyard. So what happened in that area? If we give a look to these two sites, they are located in the same soil homogeneous area, Lecce 2, and but Bro 11 is quite healthy. It don't show display any significant problem, but Bro 12 as some plants are weak, so it is possible to observe some problems due to biotic or abiotic stress. So this vineyard appeared to be more stressed, under stress, compared to the the other one, the Bro 11, but this vineyard is the one who provides the best wine quality. So which is the player, the main player driving this kind of results? So we decided to explore better the soil properties in terms of chemical, physical homogeneity of soils by using a number of different sensors, and we collected the soils again, considering the soil heterogeneity, so in order to reduce as much as possible the spatial variability of soils. And we use a metagenomic approach by means of geochip analysis, which is a microarray approach based on a number of different genes belonging to several functional groups, including taxonomy, of course, but also including carbon cycling, nitrogen cycling, etc., but also including energy processes, stress responses, and so on. So really, this approach can give you a very quick and reliable information regarding the soil process occurring in your soil sample. And the results showed a significant higher number of genes detected in the Bro 11 soils, which I remind you is the sort of control without any apparent stress, okay? Whereas the Bro 12, with some weak and some problems, displayed a lower number of genes. And this result may be related to the different content in terms of organic matter of the two soils, because the two soils are similar, but they're not exactly identical. So despite the difference in terms of organic content is not statistically significant, maybe the higher content of organic matter in the Bro 11 might justify the higher amount of microbial biomass and gene number. Okay, so I expect a higher diversity or function in Bro 11, and that's exactly what we obtain for all the genetic and gene categories, except for the sulfur cycling, where an unexpected enrichment of sulfur oxidation genes occurred. And this was quite surprising for us, but it might potentially explain the different wine quality provided by the two vineyards, let's say, because sulfur is directly involved in the together with nitrogen and other nutrients, of course, with the production of the reduced glutathione in plant tissues, and these molecules are really important in defining the wine traits and aroma of musts and wines. So this is just to let you understand that also the functional biodiversity of soils should be addressed when we talk about defining soil biodiversity in general. So this is were just an example, a couple of examples regarding this kind of approach in order to move the agricultural systems towards a much sustainable agricultural management. But the other approach is based on microbiomes. As you know, we can use microbes directly as cells, but also as molecules extracted from cells or cell suspension in order to modify specific soil processes or enhance, for example, nitrification, denitrification, or try to modify some specific process occurring in soil. But it's really common also the use of specific soil organisms as plant growth promoting bacteria, entomopathogenic organisms, and mycorrhiza, of course. So in both cases, the first approach and the second approach, the keywords to be considered are crops, soil, and microbiomes. They are the main actors of any kind of approach we decide to adopt in order to improve soil sustainability. And the interesting thing regarding this approach based on microbiomes and microbes is that, for example, a number of private companies and producers of bacterial or microbial-based inocula have developed a number of different products. And as you know, in literature, there are a huge amount of microbes available, beneficial microbes that have been studied and characterized to promote crop yield, for biofertilization, for biocontrol purposes. But the question is, why just few of them have been developed and as commercial product? Well, the companies they have, which are involved in this kind of market, when they make their own trials, preliminary trials, they obtain usually very, very nice results under protected conditions in lab scale experiment or pod scale experiment. But when they try their products in the field under real conditions, very often the results are not so efficient, or sometimes are completely disappeared. So this is a problem for the companies and for the fertilizer industry, of course, but also for the farmers, for agriculture in general, because if you take into account the general legislative framework, which induce farmers to reduce the use of chemicals and pesticides, we have to try to move forward a much more sustainable agriculture. So it will be important to have alternatives in terms of biofertilizers, bio control products. So the bio-based industry should be the main actor in this field. So what are they doing to try to fix this problem? First of all, they try to improve the formulation of the products in order to better protect the inocula in the soil, or to improve the storage viability. Another point another challenge is to try to improve and enhance the strain competition compared to the native soil biodiversity, and also to try to reduce the depredation by the macrofauna and other biotic organisms in the soil. So my conclusions of this first part are this. So first of all, I'd like to give you this message that soil biodiversity of degraded areas and soils may be restored for sure. Yeah, it might take time, decades, but it's possible. But it's impossible also to consider to ask how plants interact with soil native microbiota and the overall soil biota, and how plants may be optimized to be productive on such high diversity soils. So I invite you to consider also biodiversity. That biodiversity should be optimized with crop traits, which are supposed to be able to handle such soils. Okay, so we are also saw that restoration of soil bacterial diversity is for sure affected by soil organic matter, but maybe both, of course, by the soil organic matter quantity, but also by its quality, and sometimes more affected by soil organic matter quality rather than its amount. So it's variable, and it's also variable the approach, the soil management approach to be adopted, because in this case we saw that in our case study we saw that green manure was the best solution, but sometimes it's not. So it depends on the interaction of the proposal management with the soil, with the with the environment, with the soil biota, etc. I also invite you to consider the importance of functional diversity rather than just the genetic and taxonomic diversity, and also to have an overview, an idea of which are the limitations, but also the potentials of microbial products, or microbial-based inoculants, biofertilizers, and for biocontrol purposes. So this was the base, the baseline, the starting point of our Excalibur project, because based on these considerations, I hope at least that you get this message that we need to better understand what's going on below ground, which are the interactions occurring among plant, soil, microbes, the interactions in terms of co-occurrence, but also in functional terms. And yeah, this is why we proposed this project, and fortunately was also successfully approved, aiming to exploiting the potential of below ground biodiversity, native biodiversity in horticultural farming. And we try to stimulate, to enhance such native biodiversity by using microbial-based products, so inocula, which are already known to be efficient for biocontrol and biofertilization. So we selected 3 model crops: tomato, apple, and strawberry across Europe under both conventional and organic management systems. We try to stimulate the native biodiversity with this inocula. So this is a general project overview concept in order to make you understand how we intend to proceed. So the project started last year in June. So we have just accomplished the first year of the project. So the project lasts 5 years. So we are still in the first preliminary phase, but our intention is the overall concept starts from here, from soil. Okay, so we selected a number of soils, agricultural soils, displaying some sort of biotic or abiotic stress. So we need to start from a degraded soil. Soils under stress with a low yield, with a low fertility, low biodiversity, and we need, in order to select these soils, we of course we involve the farmers, and we selected the best places according with them. Depending on the kind of biotic or abiotic stress, we selected specific microbial strains, bacterial or fungal strains, which are already known, which sometimes also commercial products, but usually specific strains owned by the partners' collections. So these strains have been used since long time periods, so they are very well characterized, very well known. So we know that they are efficient in responding to fix some specific problem. So this is why we in this phase we try to select the best strain for the specific problem, and we provide this material, both soils and microbial strains, to the industrial partners in order to develop and set up a new formulation of such products. They will be selected based on their efficacy, of course, but consider a number of characteristics. And finally, we will select 6 new bioproducts, 3 solid formulates and 3 liquids, to be tested under real conditions, under open field trials. So the crop trials will be monitored over time, over 3 years, both in terms of agronomical response. So we will check for the yield production, the quality of the the results of the products and so on. But of course, we will give a look at constant monitoring of the soil biodiversity dynamics, what's going on over time in such soils. So in some specific, the best cases where the difference among what is going on pot scale experiments in protected conditions, where the efficiency of these products is evident, compared to the field, we will compare the different responses of the same soils under protected conditions and field conditions in order to understand which are the main and key processes responsible of a specific crop response. And this kind of information will be useful both for industrial partners, of course, because this way they might keeping on improving the formulation process of their products. But also, this information would be useful to provide guidelines and tools, practical information, and advices for the end user, which is the farmer, of course. This is the ambition of the project, because with this information, the farmers will be able to adjust their soil management practices according to the soil biodiversity driven information. Okay. So the consortium is not very, very big, but includes a number of partners from almost all Europe. It includes research partners, university, and so on, but also farmers and advisors, manufacturers, producers, and also consultants. So we try to set up a multi-actor approach team. Okay, and the experimental areas were selected with according to the farmers, but also considering 3 different paleoclimatic areas across Europe: the Atlantic Central and the north of Europe, and the continental one, and the Mediterranean, even if the Mediterranean is north, because we have not any experimental sites in the south of Italy. And the experiments were conducted, as I said before, under different management systems, organic and integrated. And some of them are conducted under open field, and some of them under protected conditions, some of them both when possible. And we try to include these 3 model crops in order to have annual crops, perennial crops, and herbaceous plants, and apple, for example, tree plants. So different kind of situations. And the main objectives of the project are, first of all, as I said, to get new knowledge of what's going on the interactions occurring among plants, soil, and microbes, but not just only microbes, but also including the rest of biota, including mesofauna, microorganisms. So we have partners dealing with mesofauna, colembula, acarina, and so on, but also earthworms, protozoa. So we try to include a different taxonomic range of soil organisms in order to have. We had to exclude. We did not involve any expert in virus, because it was too much. But we are trying to set up some collaboration with colleagues expert in virus, soil viruses. So the second aim is, of course, to optimize the formulation of the products in order to optimize the response of the use of this inoculant in the field, the response of the plant in terms of nutrition and protection. It is really important to observe and to get useful information under open field conditions, for the reason I mentioned before. And another objective, very, very challenging, is the delivery of models and tools, DSS, for practical application of the biodiversity-driven strategy for soil management. So we aim to to set up a model of the overall soil biodiversity dynamic involving partners which have experience in this, merging different public and private databases and integrating our results in these models, and try to validate these models in the field with our results and making some kind of application for smartphones available for farmers and stakeholders, of course. And another important issue is and goal is the development of tools for the detection and monitoring of the persistence and the fate of such microbial-based products of the microbial inocula in the field. So, because we can, it's important to have to have the possibility to check what's going on, where the inocula is going, how it lasts in the soil matrix, and so on. And this is very important in terms of legal support and legislative framework for the European Commission. And and the last objectives are, of course, related to the evaluation of the effects of this strategy in terms of soil quality and ecosystem functions. So we will consider also chemical, physical, biochemical properties of soils and ecosystem functions. Okay, and also the response of plants in terms of production, the quality of the products, the physiological response, of the plants, and so on. So we tried to include a really integrated approach to this project. And finally, the ambition is to provide, to deliver some very useful and practice guideline for the users. Okay, because some theoretically useful information are not useful at all for the farmers. They need some practical information. They need to see what's going on, to see if their practices are efficient or not, if it works or not, what they are supposed to do, and which is the cost of such approaches. So, of course, we also consider the economical impact of these approaches. In order to assess the soil-plant microbe interactions of the case studies I mentioned, we will, as I briefly mentioned, we will use a number of current and normal approaches based on the NGS approach, quantitative PCR, so a number of molecular tools to assess the composition of the soil microbiota and biota in the soil, but also traditional methods to assess arthropods, earthworms, nematodes, of course, protists, the expression of key functional genes related to the main nutrient cycling and soil processes, but also including the analysis of soil structure and nutrient availability and interaction with minerals. So I don't know if I'm in time, but in a couple of minutes, I just want you to highlight the importance of the delivery of these guidelines, because actually in the policy context, there's a bug, there's an empty gap regarding how to analyze, validate, register such bioproducts for commercial purposes. And so the Excalibur project also aims to support the development of legal provisions for the implementation of such products in agriculture, both for organic and also integrated horticulture, but in my opinion, it would be really important for organic agriculture. Just a few words describing what happened during the last year. So, of course, we made a number of meetings. We disseminated the project across local, national, and international events. we started with preliminary tests under pot scale conditions and greenhouse conditions. Unfortunately, we have to stop some lab work due to the COVID outbreak, but we had time to analyze, made a pedological survey of the experimental sites. We defined the main soil pedological, chemical, physical properties of all the experimental sites included in the project. We had to stop, as I said, because of the COVID outbreak, but we could reschedule, modify the scheduling of the expected activities within the project duration. So we are going to start again in these weeks with all the restrictions that we know, but finally we can start again. And we also collaborate with other similar or twin brothers projects financed and approved last year or in the last period. They are ongoing projects. So we are open to new collaborations, and we are trying to make a nice network, a collaborative network, in order to be able and be ready to fight this challenge for getting and obtain a more sustainable agriculture. So thank you for your attention. I invite you to visit our website for more information, and don't hesitate to ask questions. So don't be shy. Thank you very much.
Thank you, Stefano. Thanks very much for this interesting presentation, full of ideas, and I find also from some aspects very different from what other groups are doing. You know, you are questioning the use of this biostimulant, and this is, I find, a very interesting point, because they are not the panacea of all degradation of the soil. And so thanks very much. And it's time for question. As Stefano told, don't be shy. And if somebody would like to make questions in Italian, it is possible. Stefano understands Italian, and he will reply in English. But please, there are questions. You can raise your hand or write the question in the chat as you like. Let me see if there is some. Okay, Fabrizio De Cesare, please. Fabrizio, do you hear us?
Yeah. Hi, Stefano. Nice to see you.
Ciao.
It was a pity we could not meet in General Assembly in EGU this year, physically. Anyway, just a couple of questions, but very simple. In one of your slides, you show it. It was referred to the first group of experiment. I mean, it was referred to the different inputs of organic amendments, like green manure, compost, and some other. I don't remember specifically. But you displayed a graph where it was reported. Something like a relative improvement. I don't remember exactly, but it was just a question. You mentioned relative to what? Relative to the beginning of the experiment, or relative to, because the control were already included, so it was not referred relative. Then, for example, addition of green manure relative to the control, or relative to the degraded soil. So I suppose it was relative the last year relative to the starting of the experiment. Is it correct?
Yeah,
Okay.
you're right. I apologize for not being so clear. Yeah, I showed the relative increase in terms of total carbon and nitrogen amount after 2 years since the beginning of the experiment. So yeah, so it's
Okay.
compared to that.
And when you talk in several slides as a conclusions of the different steps of your absolutely fascinating information you provide in this presentation, and you talk about functional diversity. So is it something, when you talk about functional diversity, is it something that we can refer to what Professor Tebbe talked yesterday in the interactions within the biodiversity and the very spread different and significantly different interactions within microaggregates? Can we refer this functional diversity to the diversity we can find inside the microaggregates? I mean, in terms of interactions within different groups of microorganisms.
Yeah, so that's a nice question. I mean, yeah, as Professor Tebbe showed yesterday, the fractionation of soil aggregates is really important, and different fractions provide different microbial communities, bacteria, fungi, etc., and also functions. In our case, the case study I showed that we did not take into account such fractions. So we just check the overall response of the soil sample of the two vineyards in terms of metagenomic response and enzymatic activity, soil microbial respiration, and I assume we also consider the importance of different fractions. But in that case study, we compare two similar soils, so they are almost identical in terms of soil texture, aggregate composition, and structuring. So we didn't believe that such kind of variable might be relevant for assessing the functions in that specific soil sampling, but it's just related to that specific case study. In general, yeah, you're right. As Professor Tebbe said yesterday, we should pay more attention in what are we looking for, and which is the right way to collect samples, and in terms also soil fractions. So I don't know if I answered.
Okay, thank you. Yeah. Nice. Okay. Thank you very much. Just close. I'm working on biostimulants or something that tends to be a biostimulant.
We need to meet each other
I
in
will
person
contact you
soon.
privately.
Okay. Thanks Fabrizio. Thanks for questions. Thanks very much. Can you put down your hand so I can see if somebody else? Okay, thanks. Are there other questions for Stefano? So don't be shy. Oh, okay, Luca Giuga. Please, Luca.
Buongiorno, farò la mia domanda in italiano direttamente. Okay. Si è parlato durante la presentazione di vari aspetti molto interessanti, ma molto più complessi da indagare. Gruppi funzionali, interazioni biotiche e via discorrendo. Attualmente noi non conosciamo circa il 90% delle strategie alimentari del biota del suolo. Abbiamo una conoscenza frammentaria e limitata sulle interazioni, insomma sappiamo che siamo molto indietro per ciò che concerne lo studio della fauna del suolo sensulato. Hai parlato di una sorta di monitoraggio, di valutazione che include vari gruppi funzionali, quindi si è parlato di microbiota, di collemboli, acari e via discorrendo. Attualmente un metodo standard per la misurazione della diversità del suolo non esiste, che io sappia, per tutta una serie di ragioni, time spending, mancanza di esperti, via discorrendo. Ora mi chiedevo, visto che illustravi questo approccio che include vari gruppi funzionali, se non tutti, come pensate di superare questo limite? Grazie.
Okay. I'll try to translate, summarize the question. Please correct me if I'm wrong. So Luca said that, yeah, we know we have poor knowledge about what is occurring below ground in terms of soil biodiversity and the interaction among biota organisms. And actually, a general method for assessing the overall soil biodiversity is not available for different reasons. And he asked how we are going to face such problem, such complexity in our approach proposed in the project. I mean, right, Luca?
Yes.
Okay.
Yes.
So yeah, nice question. So that's the point. Yes, you're right. There's actually not a common and general strategy or method to assess soil biodiversity for a number of reasons, and I'm not expecting that it would be developed, but it's important to understand, to use another perspective. I mean, I try to show you, asking several questions, because I'd like to stimulate your critical approach, okay, and to think about the problems, the real problems. So soil is really complex, and the interactions are really complex in terms of spatial heterogeneity, but also temporal spatial variability. So what I saw now, what I'm able to measure in this moment might be different from what I can measure tomorrow, or in the next few hours, or next week, or next month. So it's really complex. And so this is why we try to say, okay, let's start from something that we know. So we have isolated strains that were shown to be effective in promoting plant growth or suppress a specific pathogen, etc. But when I use this strain in the field, it doesn't work anymore. So which is the difference? In the difference, which are the main parameters driving this different response? This is the strategy. So we use the inocula as starter, like a stimulation of the native microbial community, native biota in the soil, to check what's going on. Which are the trophic interactions, the nutrient processes, the functions involved in the response of plant or in the missing response? Because from one side, I see the response in the pot scale with that soil. The same soil in the field maybe give us a different response in terms of plant yield, plant health, etc. So comparing the two situations, we aim to get such information, to understand what's going on, why this plant in this situation responded differently from what I saw in my lab or in the pot. So we are really curious to see what's going on, because we can find the same organisms, the same biodiversity, but different expression of their functions, or different interactions, different actors, or whatever, or different response of plants, or different metabolic profiles of the microbial communities. So our intention is we decided to approach the problem in this way because we are aware, we are sure that we can't analyze, try to get a sustainable agriculture just with plant breeding or improving plant diversity, or just focusing our attention on soil management. Is plant soil microbes is a unique system. So we need to approach the evaluation of the functioning and the efficacy of this system with a very multidisciplinary approach, considering all the variables, at least, as much as possible. But that's the challenge. I don't know if I answered to your question.
Very interesting. I don't know,
Yes,
Luca,
yes,
are you satisfied, Luca?
Luca, se non hai capito la risposta, poi te la ripeto in italiano.
No, no.
Non so se posso rispondere anche in italiano, ma poi no.
No, è tutto chiaro. No, è molto chiaro. La mia non era una critica all'approccio, cercava di indagare il come pensavate di ottenere questo risultato, conoscendo la complessità che sta alle spalle di questo.
Comunque
Grazie.
mi potete anche contattare singolarmente per eventuali curiosità o informazioni aggiuntive. So, in case you have specific questions or curiosities, please feel free to contact me by mail to ask questions. So.
Okay, thank you.
Thank you, Luca. Thank you for your question. Put down your hand, please. Put down your hand. Are there other questions to Stefano?
There is one question in messages.
Okay. Let me see in chat. Shamina, do you want to make the question to Stefano? Do you want we read your question? Can you hear us?
Yeah, I can hear you.
Can you make the question directly?
Okay. Hi, Stefano. It's a nice presentation. I would like to ask a question related to the techniques, because in one case study you use the microarray, and in the new project you saying that you will do the amplicon sequencing and gene expression RT-qPCR. Yeah. So can you put some lights on pros and cons for different techniques, because somehow you will have some differences in the results if you use the different techniques.
Okay. Yeah. Thank you for your question. Yes, I shown in my case study. In that case, we used the geochip approach, a microarray, rather than NGS, because as you know, there are different kind of molecular approaches. Let's say that we can divide the molecular approaches in open and closed approach. The closed approaches, including microarrays, kilochip, geochip, and so on, relies on the question, on the aim you have, because you have a system that already include a number of known genes on the chip, of course. So they are, at least I recommend to use this approach when you just need to compare two different situations regarding some specific gene function. So, for example, in the case of Broglio, we used geochip because we wanted to check if some different gene detection in terms of gene functionality was occurred among that sites. But the NGS, the metagenomic approach, is considered as open approach, because when you sequence the overall DNA from some sample, you explore what's in that sample. So you are able to detect also unknown molecules, unknown genes, unknown functions, and it's really it's much more informative in this way, but it's also much more complex and less reliable as functional, in terms of functional information, because there are several critical steps along the pipeline. So in that case, we prefer to use GeoChip for just comparing two situations that we know for specific reasons. But in general, like in the case of the Excalibur project, where we need to explore something that we don't know. We need to find out answers to our questions. We need to find out any possible parameter, function, organism, or expressed gene that may be involved in the different response of that soil. So this is why we propose to use NGS, metagenomic approach, and also transcriptomics, of course, both for plant expression, but also for soil. But as you know, these approaches are quite expensive as well. So we will apply such approaches just on the selected cases. The more efficient, more interesting cases we will observe during our project. But in the beginning, during the 3 years of monitoring, we need to analyze a huge number of samples. We decided to use normal, current, ordinary approaches, so microbiome barcode-based analysis, and so on. But yeah, you're right that there's a number of different options in front of us, but we can, like Tebe said yesterday, we should select the best way to sample soil depending on what you are looking for. So the same thing I said when you have to select not just the kit for DNA extraction, but also the technique to analyze the DNA. So you can use an open approach like NGS or metagenomics and so on, or also a closed approach for different purposes. And both have potential advantages and disadvantages, but both really interesting. And regarding the second question is much more related to the active community. Yeah, we also consider the RNA approach, of course, as I said, in order to catch such community fraction. But as I said before, we just limited such approach for just some selected cases, not in general.
Okay, I have one more question.
Shamina, sorry, I don't know if I see also Eduardo, because it's almost 10:30. We have another lecture in the school. Yeah. But Shamina, you can contact or write Dr. Mocali. Yes. And I'm sure that he will answer your question. Sorry very much, Shamina, to stop your further question. So I would like to thank Stefano for this interesting talk, and also for the so complete answer he gave to the questions made. And again, we understood that how complex, dynamic, heterogeneous is the soil-plant microbe system, and how much is important again to stress the necessity to upscale results obtained in controlled environment. Because out in the field, sometimes we look different results. Maybe something we do not expect, you know, and this is a very challenging, I find. Upselling is very challenging part of the research. So thanks very much, Stefano, to have been with us, and now we close the general channel, and we move with the students of the school on the private channel for the lectures planned of the school. Thanks to everyone attending this first part of our school and the public seminar. Bye.